Welcome to another episode of the Bioinformatics Chat. I'm Jacob Schreiber, and I'm joined today by Dr. Žiga Avsec. Žiga did his PhD at the Technical University of Munich under Dr. Julian Gaggenau. Now he's a research scientist at DeepMind. Thank you for being with us, Žiga.
Yeah, thanks for having me.
So what's it like working at DeepMind? It seems like it's a pretty popular company. It's always in the news. I imagine that you have a good experience there.
Yeah, I like it. It's a really great environment to do research. And yeah, there's a lot of exciting things going on at DeepMind and lots of, great people to talk to and brainstorm about ideas. And yeah, so I really like it.
One of the things that I hear a lot is that there's kind of a distinction between doing research in an academic setting, where you kind of have your freedom to pursue whatever you want, and then industry jobs, where, if you're in academia, then industry jobs are kind of like looked down on. But then more recently, it seems as though there has been a lot of kind of blurring of the line, where there are really great industry jobs that involve doing cutting edge research, and that DeepMind is one of the places where this seems like a possibility. Is that something that you have found?
Yeah. I mean, we like to say at DeepMind that we're kind of doing the best of both worlds. Although, I mean, this is, of course, you have to take it with a grain of salt. To have the freedom of academia to explore really fundamental questions in science and try to address them and be really ambitious. And on the other hand, to still be focused and to have that industry focus on problems and be able to kind of sustain that focus across even multiple years, for example. So I guess that's, kind of to me, the biggest distinction with academia is that, you do less things, but you try to do, you try to be more focused on specific problems. But the problems that, the questions that we're after, at least as part of the science team at DeepMind, are just generally open questions in science. So it's pretty similar from that perspective.
I think that one of the differences is kind of the unit of focus, in the sense that when you're in academia, a lot of focus is put on the next publication. And so you want to know how much you need to do in order to, finish that next publication, then move on to another project. Sometimes in industry, I'm going to ask you whether or not this is the case at DeepMind. Basically, the unit of focus is on some greater product, where you care a lot more about, like in the case of AlphaFold, like the higher problem is trying to figure out like protein folding. And if a manuscript happens to fall out, that's great. Otherwise, you're still trying to focus on this larger goal. Have you found that in your work generally that the focus is less on publications?
Yeah. There's not so much pressure to publish to then get tenured and so on, because of the the recognition system is slightly different, I'd say, from academia, where academia is primarily measured by your publications and then your grants and so on, whereas in DeepMind you kind of are already in an environment, and that kind of allows you to focus more on maybe more long-term goals, I would say, and then really try to think on where you're trying to get to overall, and then think about the publications as a secondary step. Although, I mean, it's very important to us to still publish really good research, because obviously you also don't want to postpone for too long, because you want to, allow other people to also build on top of your ideas.
Right, of course. I mean, some could also say, though, that there's less focus on, tenure, because you don't have tenure at DeepMind. That's, an academic construct.
Yeah, exactly, yeah.
So we invited you on the show because we had you recently had a publication. I think that it's basically the blueprint for how computational papers in biology should be performed. First, burn out of academia and go get an industry job. No, just kidding. I really enjoyed reading it, but when I was talking with other people about it, there was a huge criticism that almost always came up, and I was hoping that you could address it first. Basically, this work kind of grew out of the work that David Kelly did with Bassett and Basenji, where the idea was you were taking a nucleotide sequence and you were trying to make predictions for various tracks. Now, this paper proposes a model named Enformer. And already right there, like, you are deviating from David Kelly's naming scheme of focusing on dog names. So I was hoping that you could clear the air and tell us why you chose this name Enformer as opposed to, say, Corgi.
Yeah, we had a long discussion around how we should name it, and yeah, I don't know. At the end, you just have to pick one. We couldn't come up with a good dog name, and we also have some people that really like cats at DeepMind. So no, I'm just kidding.
I guess that one of the downside of trying to pick a dog name is that because the model relies on like long-term attention, there's no real dog that kind of fits that mold, right?
Yeah, probably. Although I have to say, we searched a bit for dog names, and then yeah, if you go into the regime of these big dogs, then they kind of have this dangerous connotation. So you also don't want to have something that looks super dangerous. So I think maybe that's why we kind of deviated from that point.
Yeah. So you started working on this project, this Enformer work, kind of off the heels of this paper where you proposed this model named BPNet. Can you talk a little bit about your inspirations for BPNet, give a little bit of a description of what that is, and then talk about how that fed into the Enformer work?
So BPNet is kind of a smaller and more focused version of a similar kinds of architecture, I would say. So BPNet is also a model that takes as input DNA sequence and predicts the experimental coverage tracks. In our case, for the paper, this was ChIP-nexus data, which is basically high-resolution ChIP-seq data that measures transcription factor binding occupancy in the genome. And the key feature of this data set is that it's high-resolution, so you get at the location of where transcription factors bind in the genome, you get markant footprints or signatures where you see really high amounts of reads and the key feature is that they are really precisely placed, so you can maybe even get down to a specific base nucleotide that is at the boundary of the transcription factor.
So you're talking specifically about the fact that the data is from ChIP-exo as opposed to ChIP-seq?
Exactly, yes. Exactly.
Okay.
So the key motivation there is basically what model or loss function should you use to learn from this data, as well as then how to interpret this model in more detail. And in that project, we used dilated convolutions and a specific loss function that was able to deal with this very sparse ChIP-nexus data. And the model basically is, tries to recreate the experimental signal. And by doing so, it learns about the binding of different transcription factors. And then we had a series of interpretation tools that were then able to distill the knowledge that this model has learned into objects that we're very familiar with. In this case, these were transcription factor binding motifs, as frequently used in the bioinformatics community, as well as to study the spatial relationships between the binding of transcription factors. So, for example, does a transcription factor bind more to DNA if you have another protein nearby to it, and at what distance should they be apart in order for that binding to be most highly effective? So in relation to the Enformer work, you can think of the Enformer as or also the Basenji work done by David, as a scale-up version of it, where BPNet is trained on 1,000 base pairs and it outputs data at base resolution, so it will predict a signal at every base. Whereas Enformer and Basenji is trained, the input is 200,000 base pairs, so it's 200 times longer input sequence. And the output is not at base resolution, but it's at 28 base pair resolution. So because it becomes quite challenging if you're dealing with base resolution data at such long distances. It just burns a lot of memory. So the knowledge is that they're both sequence to sequence models. They predict experimental data, but they do it at different scales, and therefore they capture slightly different biological phenomena. So BPNet captures more the binding and the interaction between individual transcription factors, whereas Enformer tries to focus on higher order molecular phenotypes such as gene expression, and how this is composed out of more basic units such as DNA accessibility and so forth, to arrive at then accurate predictions.
Yeah, I think that's a really good description. That the general trend I've seen is that the models like Basenji, Enformer, etc., although they do take in nucleotide sequence to try to predict some sort of functional track, that they're doing it at a much coarser resolution, and so you're able to pick up these much larger things, whereas a model like your previous work with BPNet focused very specifically on fine-grained detail, base-level details, and the insights that you can get when you look at changes at single base pair resolution.
Exactly, yes, exactly.
So what inspired you to start this work on Enformer, to basically take the Basenji model and say, this isn't good enough, and continue the work on it?
It's clear that so there's been several papers now on published, and we know a lot, but not yet as much as we would want, about enhancer biology and about at what long distances do enhancers. So enhancers are regulatory elements that typically attract transcriptional machinery, and they enhance transcription of nearby genes. And it's known that so the Basenji model had a receptive field. So this is basically whenever you're predicting specific position in the genome, is how far left and right of that position will you see, like how much of the sequence will you see left and right of it in order to make your prediction. And in the case of Basenji, I think this was 20,000 base pairs, I think, left and right. So 40,000, the receptive field is 40,000 base pairs overall. And we know that you can have enhancers that act at much longer distances. You can have even things beyond a megabase. So it was clear to me that you need a better way to capture these long-range interactions. Although one easy thing you can imagine, is to basically just put more layers into Basenji and gain a larger receptive field that way. But the problem with that is that the path between long-range interactions and your position of interest that you're trying to predict wouldn't be very direct. And at some point, if you're using dilated convolutions with a width of 3, the next bin starts to reach out of the sequence, and it's not as meaningful anymore. So I saw the need to reflect the biology better in the model architecture, and transformers seem to quite a good fit, because they have this ability to basically, the connection between an element and another element in the sequence that you're trying to look at is direct, right? You don't have to integrate that information over many steps. So they seem like a good fit.
So to summarize, basically you saw that Basenji had a limited receptive field that was much smaller than you expected real biology to be.
Exactly.
Because these regulatory elements can act over very long distances. And so you said, rather than having multiple layers of dilated convolutions, increasing the receptive field sequentially, like Basenji does, you were saying, what if instead we just used an approach where you model all pairwise contacts, like a transformer?
Exactly, yes, exactly.
Yeah, so I thought that made a lot of sense to me. That kind of with these dilated convolutions that you were, it doesn't seem super reasonable that you need all of these layers in order to do very complicated processing of the sequence in order to get the output. It seemed more like the multiple layers were necessary just to slowly increase the receptive field. But if you were able to replace that whole mechanism with more powerful layers that were they were aggregating information from everywhere at every step, that you could learn a more powerful representation.
Yeah, exactly.
So do you want to walk through some of the basic results that you saw? I assume you compared to Basenji and saw that Basenji did a lot better than you, and so you had to be like, oh shit, and then throw out those results real quick.
Yeah. I mean, the road towards like getting the model to work isn't straight always, but yeah. Um, it took some time to get the model off the ground, especially with transformers. You need a few tricks to get them to work. So I was really fortunate to be at DeepMind and be really surrounded with people who are experts in getting these models to work and to have all the tricks at hand.
That's actually a really good point. I was maybe you're about to talk about this, but I was wondering if you could talk about some of the tricks that you got to work. You talk about them in the paper.
Yeah, yeah. So one of the things was important was, for example, like increasing the learning rate, like gradually increasing the learning rate, and I think this was probably one of the most important things. And then putting the normalization right before the attention was also another trick that allowed us to fit this data better. And just generally, yeah, you had to do a bit of learning. The learning rate is much like it's quite different from Basenji. So it seems like transformers need much lower learning rates compared to dilated convolution. So that also had to be tweaked um, to get it to work, basically.
Yeah, yeah, but these these are the tweaks. These are the boring things. I was talking about these basis functions that you were using.
Oh yeah, this was another thing. Yeah, maybe let me just explain briefly about how like how these positional encodings work in transformers for context. So there are two ways to encode positional information into transformers. You can do absolute positional encodings where you add you basically you have different sine waves like basis functions that, and then you add slightly different signal to different positions, so that whenever you're looking at two different positions, you're able to roughly tell how far apart they are by basically kind of like unpacking that extra component that you added in at the beginning. And they're called absolute positional encodings because this is what you do something at the beginning, and then you let the model like learn about this. The other and this works pretty well in language. The other category are relative positional encodings, where when you're building up the attention matrix, where you're basically computing how much each position should attend to other positions, you are also considering the relative distance. And then you have a separate function that tells you, okay, if you're, I don't know, 10 positions apart, you should prefer positions more at the specific distances. And you can also have terms that, depending on the query kind of content, you can look for different distances. So for example, if you're, I don't know, an enhancer, then you might look at more proximal things, whereas if you're a promoter, you might look at more distal things.
So to jump in and to summarize real quick, that the transformers by themselves have no notions of positions. They take in sets of elements, and the self-similarity doesn't account for the position in the sequence. And so when you're in domains like NLP or whatever, where position does matter, what they do is they concatenate features to the representations based on the either absolute or relative position of those features to each other. And so when you tried these kind of traditional position embedding approaches for this genomic data, you found that it didn't work particularly well.
Exactly. Yeah. So with these relative positional encodings, either absolute or basic positional encodings of basic relative positional encodings, we couldn't improve it beyond beyond dilated convolutions. And then I started thinking on like what would be a better way to model this, and what are kind of the key properties of this data. And one of the key features is that there's kind of a power law decay of the influence between enhancers and promoters. So I thought maybe we have to put something like this into the model, or we should give the model the ability to learn about this. So yeah, and then we added a special set of basis functions that are closer to what we expect to happen biologically, to have these decaying functions, as well as we also added the asymmetry to basically distinguish between what is upstream and what is downstream of the elements.
I think that makes those made a lot of sense to me, that you would try to incorporate these types of biological constraints. I think one of the big differences that people sometimes they see things that work well in the NLP setting and they say, oh, that's just sequences of words, right? And then they see genomics like, oh, that's just sequences of amino acids or nucleotides, so they must be the same thing. But really, they're quite different. That with language, you can have a single word that, regardless of where it is in the sentence, well, not regardless, but there can be a single word anywhere in the sentence that can drastically change the meaning. Although you can have variance in, genomic data, typically things are a lot smoother. That you need, entire regulatory elements, and like you're saying, there are these physical constraints that even though linear distance doesn't quite mean distance in 3D space, it sort of does.
Yeah, exactly. Yeah, the language is much more structured than DNA, right? The DNA is kind of evolution gradually adding up elements, and then yeah, it's a different construct a bit.
Something though that kind of was inspired from the NLP field that I was thinking about is that typically people, when they talk about like relative encodings, they would look at the distance in the sentence. And that is kind of a naive approach that, of course, words that are closer together probably have more similar, they're operating on the same meaning. But in truth, what you want to operate it on is like a parse tree. You want to have relative embeddings where the relative encoding comes from distance along the parse tree so that you can get things that are in the same clause to mean the same thing, regardless of, if you're crossing a comma or whatever. And so in the same way that one might build a relative embedding based on a parse tree, it seems like potentially you can encode distance here, relative distance, using a Hi-C contact map. I was wondering whether or not you considered trying to use chromatin architecture when creating these positional embeddings.
Yeah, it's an idea that came up as well. Although for this project we wanted to stay we wanted to use it we wanted to have the model to be sequence only. So we went down that route to basically keep it sequence only, and then try to see if there's any properties that the model has learned that are reminiscent of things like contact maps. And we saw some, for example, like insulator elements. Although it's still unclear, as you said, with the parse tree and so on, if this is the optimal architecture to basically say on whether you're looking beyond a specific insulator element or not, because that information has to be integrated across multiple layers in the architecture.
That makes sense. So one of the reasons that I really like this paper, and I think it's kind of the standard for how computational modeling should be done, is that you start off the paper, and of course you talk about the related work that you build upon these previous models, and you show then the generic setting of just you train on some chromosomes, you make predictions for other chromosomes, that your model outperforms these previous approaches. And you have a lot of cool visualizations showing not just global measures, but also visually, like, look at it, it's better. I think that is all particularly compelling, but that I think that sometimes a lot of papers in computational biology stop there. They say, okay, my model is better, therefore one would use it in theory to do all of these other things. But for your paper, like, you're done with all that by like page 3. That most of the paper is spent on, okay, we have this better model. What does that actually mean practically? And then you go and do these things. You were just talking about how one of the things was that you wanted to see, like, are you able to, based on these predictions, are you able to do things like uncover chromatin architecture? One of the things I found is particularly interesting was that you tried looking at whether or not the saliency from the model was able to uncover enhancers. Can you talk a little bit about that?
Yeah. So first off, really good observations. When building sequence like DNA sequence models, like the classical evaluation is across chromosomes, where you train on a few chromosomes and test on a few other chromosomes. But yeah, as you said, this is not the end goal in itself, because we already have the data in those chromosomes. We already measured it. So the fact that you can predict those accurately, it's nice, but yeah, you have to do other things with it. Either interpret the model or use it for other predictions, for example, to generalize to mutations. In terms of analyzing what the model has learned and whether it has focused on enhancers. And this is maybe a bit similar to BPNet, where contribution scores are really useful tool for studying, given that the model has made a specific prediction, what input features were most informative. And this is really, big kudos to Avanti, Srikumar, and Anshul's lab and so on for kind of pioneering on these methods. And there, the question is the model looking at specific regulatory elements? And then the question becomes like, what is a good benchmark for, promoter enhancer interactions? And surprisingly, it took us quite a bit of discussion on internally and what data we should use there. Fortunately, there's been really good data sets on using CRISPR-I, so inactivating chromatin using CRISPR to then see what's the impact on expression. And I think this is a very good experiment to probe it, to probe the promoter enhancer interactions. And we saw that indeed the model performed quite good on looking at enhancers. And we saw also that the good performance, you had to really look at the contribution scores with respect to the output target that was specific to that cell type. So in this case, this was K562. So if you, for example, backpropagated from an average cell type representation, then the results weren't as good because the model might have been looking at other positions that maybe the enhancers from other cell types. And so there we saw the performance was similar to another model called ABC model, so activity by contact, where the idea is to multiply the activity of enhancers, typically measured by H3K27 acetylation, by you multiply this with high C contact frequency. So yeah, it's two very different approaches. So I was really surprised to see that our model did so well on this task. Although for all practical purposes, if people want to use this, I would still recommend to use the ABC score because it's more practical. You just need to need two tracks, and it's much cheaper computationally.
I think that's also something that comes up frequently with computational approaches, that after somebody's been working on something for so long, you present all your results, and then people are like, oh, so you're going to use this in practice? And the person's like, no, of course not, because all of the all the flaws in it. But I do agree with you that these data sets are really interesting, because the data sets that you're evaluating against for determining enhancer activity, because they're measuring direct functional interactions. They're saying when we repressed this locus, that we actually saw a decrease in gene expression. And so that's a causal relationship there. And a lot of the times the data sets that we have in genomics, and also more broadly just in machine learning, are based on associations that you can't really determine causal. You can't really determine whether or not something's causal. But these data sets collected at great cost allow you to do that, and your model actually does quite well at a lot of them when only seeing sequence. It's really interesting because that's kind of the canonical thing that people want to do with these attributions. They train the model, they make the predictions, and they say, okay, well, why did you make these predictions? And like you said, that there's been a lot of work on trying to justify that from a principled standpoint. And to be able to see a model like Enformer trained just on all these various genomic tracks, and then when asked to explain its predictions, said it's actually this enhancer right here. That's the reason. That's the kind of the end goal of computational biology. That's exactly what you want. And of course, all of the predictions from the Enformer model are perfect, and there's no need to use anything else to validate them or anything. So we basically solved biology, I think.
No, yeah, we're far from this. I think biology is extremely complex, and yeah, we have a long way to go, which is also the fun part, I would say.
Yeah, no, I think that's true. I think that it's kind of similar to the ImageNet moment in computer vision, that beforehand people were just playing around with oh, can you identify a dog in this image? And it was still kind of like an open question. And then these big networks like AlexNet or DanNet, if you want to try to give credit to Schmidhuber, these big networks came out around 2012, and they started just blowing everything out of the water. And people were like, oh, is computer vision dead? But it's not. That's kind of the opposite of what happened. That it's not that computer vision died, it's that it was stuck on this very basic question for a very long time. And now that they had computational models able to solve those basic things, it's opened up being able to do so much more. And I think that here, that just like Informer and these other models are getting what seems to be state-of-the-art performance. It is state-of-the-art performance,
Yes.
but it's state-of-the-art performance are kind of a task that people have been banging away for a really long time. And now that we're able to do it, it opens up so much more complicated questions we weren't even able to tackle before.
Yeah, exactly. I think these improvements generally, I think because biology is so complex, you open up more questions. As you open up many new, or you offer like the ability to ask more questions, and therefore, yeah, it becomes really interesting.
So one of the avenues moving forward is trying to identify variant effect. And this is something that's particularly interesting to me because it's really a complex topic that we discussed this on a few of the previous episodes of this podcast, including the ones with Dave Kelly. The idea is basically that, all of us humans have hundreds of thousands or millions of mutations with respect to the reference, and some of those do nothing, and some of those end up causing a lot of disease. And it would be really convenient if all of those mutations fell in, obvious locations, like, oh, this is in a gene body and changes the amino acid from one to another. But a lot of them kind of fall in the middle of nowhere. And so a challenge for the last decade has been trying to assign meaning to these variants that kind of fall in the middle of nowhere. And you showed some of these more basic, you showed some of these results with Informer, but you also compared the result of Informer to a recent challenge, the KG5 challenge. Can you talk to us a little bit about what that challenge is and the results you saw with Informer?
Yeah, so the question of interpreting genetic variants is indeed like central to this approach, and this is kind of, I would say, one of the main motivating factors of developing these models is to be able to shed light to variants in the non-coding genome. We evaluated our performance in the paper on two different kinds of data. So both are genetic perturbations where like you can think of it as an edit is made to the DNA sequence, and then the question is how much did expression change, and and these two benchmarks were so for you were asked specifically for the last one for the KG5 data set. So there was a challenge called critical assessment of genome interpretation, where they asked participants, so they performed experiments using massively parallel reporter assay, where you basically take a sequence of a regulatory element, either enhancer or promoter, and you mutate and you generate many sequences from it by systematically mutating every position to other variants, and then you measure the expression change, and that way you kind of profile the landscape of that enhancer. And then the question was, in that challenge, can you predict the effect on expression these mutations will have? And we were very surprised to find that we actually outperformed the winning team of that challenge, which also used methods based on deep learning. So, for example, I think they integrated things like deep C scores and so on. And we saw that you can actually get really good performance by just taking Enformer off the shelf and using, I think in our case was DNA accessibility measures. It was surprising that I think accessibility worked better than CAGE, which measures expression in that case. Presumably this is because of because they measured enhancer elements, which I guess anyway, so this is still unclear on why exactly this was a better.
So just to be clear, so the goal of the challenge was to assign to each potential variant the effect that it would have on gene expression.
Exactly.
And so what you took is the attribution from the method at explaining only the DNase track for the cell type of interest.
So the Enformer model is trained to predict 5,313 different genomic tracks. 638 of them are related to CAGE, which measures gene expression or transcriptional output. And we also have some measuring DNA accessibility as DNase, and they are measured in different cell types. So in that challenge, what we knew in what cell types these experiments were performed. So we made predictions with Enformer for the reference sequence and the alternative sequence with that base mutated, the base of interest. And then we compared predictions for DNA accessibility for the genomic track DNA accessibility genomic track in this cell type of interest, and we took the difference between them. And I think we had to filter, because Enformer is obviously based on a very long sequence. So I think we only took the changes in DNA accessibility in the central region, because we kind of had to inject the sequence of that plasmid in the center of the sequence, and then make these predictions.
You're saying that because the input and the output of Enformer is so large compared to the what they're looking at in the challenge, that you kind of had to artificially restrict what Enformer looked at.
Exactly. Yes. Exactly. I think I don't remember how we padded the model exactly. I think either we did ends or just random sequences or something like this. Yeah, and then for output as well, because you could be because the only relevant output is directly on top of the regulatory element that you're studying, so we had to gate it on that as well. But it was still, it's still really surprising to see that the model was able to do so well, even though, as you said, it was only trained on the reference genome. And has never seen any variation from it, basically.
Yeah, I mean, I find it particularly surprising because the problem of variant effect is inherently out of distribution. That people sometimes think that in distribution or out of distribution just means whether or not the value for each feature has been seen in the training set. They say like, for images, that seeing entirely zeros might not have actually been seen in the training set because that would be pure black. But with DNA, we see ACGT at every position, so all of these sequences are in distribution. But the genome is complex and it's very structured. And so if you're training on the genome, it's possible that changing particular variants can make it be like something that's unseen, that's totally out of distribution. And so to see all these models, not just Enformer, but the other machine learning based models, be able to generalize like that is really interesting.
I think why this is possible is because DNA is still compositional in nature, right? You have, for example, transcription factor binding sites, which are, 10 base pairs long, let's say. And you can kind of see all 10 mers. I mean, 10 mers is maybe too much, but you can explore a large sequence space for, if you make the sequence short enough, as you said, like for a single base, you explore all the possibilities. It's easy for 2 mers as well. And then as you go, at some point, you stop. So the question is, if your biological phenomena is encoded in these short sequences, then, it's easier to learn the deviation from it because you've seen the sequence space there. The other question is then, how does the composition, have you seen enough composition of these different elements in action to be able to generalize across them? But yeah, I mean, ideally you'd want to, sample many, many different sequences. And this is also why, so we're also training on the mouse genome, which kind of acts as fresh sequences. And this was really a great idea that, David and others had for training models on other species to get extra sequences. And because, the code is, similar, it's surprisingly similar between humans and mouse. I'm always surprised when you think about a mouse and a human, that how much of the regulatory elements and circuitry is similar between the two.
I think that people have this baseline that nothing is shared across species. And so when they see that some stuff is shared, that seems like a lot. I'm not sure what a good reference actually is, though. You have the same proteins in multiple species, and it would make sense that the same proteins would interact with DNA in the same way. But kind of getting back to a previous point, that's not just, yes, of course, you've probably seen most protein binding sites in the genome, and you've probably seen a ton of different variants of that. But if you just take if you take some motif that's length 9, then that's there are 3 to the 9th possible scramblings of that, and I guess 27 different 1 edit distance scramblings of it, that it's unclear to me whether or not, because so biophysically what's happening is that when you edit one of the nucleotides, that one or more of the amino acids on the protein is no longer has as much binding strength. And so depending on how strongly that protein is binding, either it will have very little effect because other nucleotides are providing a stronger binding affinity, or it can have a huge effect because maybe that was the only thing that was causing it to hang on. But that's not the simple binding of a protein at a location isn't the whole story for regulation, which I think was a point that you are getting at with being surprised by why the mouse genome was so similar, that there's the composition of other proteins that are in the region that kind of confound all of these things. And so despite the fact that you've probably seen each protein binding site
Exactly, yes.
many times in the training data, I feel like you haven't really seen the whole environment more than once of other proteins binding in a similar region.
Yeah, I mean, the generalization capabilities are, of course, would like it to do better, of course, and this is one possible direction of improvement. But like you've seen a few combinations of transcription factors, definitely not all exhaustively. Yeah, I would say it's an open problem.
I mean, what we really need to do is that we need AlphaFold for DNA protein. I've seen people take AlphaFold and just concatenate two proteins together and fold them together. Just concatenate the DNA sequence. You can just, I know that it wasn't trained on DNA, but they're like, most of those characters, A, C, G, T, are also amino acids. So I'm sure you could just put those together and, you should be able to get the binding affinity for everything. It's deep learning, right?
Yeah, sure. I guess it's not that trivial, but it's just, yeah. I guess each of these things has its own caveats, and it's not that trivial.
So maybe you can talk a little bit about the compute requirements for a model like this. That Basenji, when we were talking with Dave, that Basenji itself was taking a lot of compute because it was this like 11 layer, 11 dilated convolution layers trained on, genome-wide on 5,000 tracks. It's kind of a monster. And now you've taken these dilated convolutions, which, while complicated, they're not nearly as compute intensive as transformer layers.
Yeah, so this is one of the big advantages of being a deep mine, is to have access to compute, and also tools to be able to run these, like train these models on hardware such as TPUs. So the Enformer model is using 64 chips, TPU v3 chips. So it's kind of equivalent, not exactly to 64 GPUs. And we're training at batch size 1 per chip, or like our batch size is 64, but it's one sequence per chip basically, because that's it's kind of the memory limit of these chips, is that as we increase the model, our memory requirements also went up, so we can only train on batch size 1 per chip. And the model trains, I forgot exactly how long it took to train, maybe 1 or 2 days or so. So it's a pretty substantial amount of time of training the model. So it's like experimenting with new architecture becomes more difficult. With VPNet, it was, for example, very easy to traverse the architecture space and try out new ideas, because the models were trained under, took a couple of minutes to train, whereas maybe up to an hour. Whereas here, if you're talking about multiple days, it becomes more challenging to try out new ideas. So you have to be a bit more careful of how you go, like what things you try out.
It did seem though like you tried out a kind of comprehensive set of ideas. You have a lot of these figures in the supplement where you compare alternate things. Was it more that once you had kind of figured out what the best general approach of model was, that you kind of fleshed out the space around that?
Yeah. For the paper, yeah, we ran a bunch of ablation studies, because like the way to getting the solution is quite messy, of like trying things out and see what works. So at the end, we wanted to, as you said, like map out the space of to see basically what really helped the model and what not, and to try to really pinpoint it at what specific aspect of it is helpful and which ones aren't, to also give some insights to the community as in what things were important and what aren't. So for example, if people want to build on top of this work, so that they would know what is, for example, not important at all.
So if you had to summarize some of those things for a general audience, what would you say generally was very helpful and generally was not helpful at all?
One thing that was helpful was these relative positional encodings. We saw a nice boost from using them compared to standard like relative positional and basic functions that use sine, cosine. We also increased the number of channels. That helped a bit. So that increased the capacity of the model. Something which was quite interesting that we saw is, for example, let's say if you restrict the receptive field of the transformer to be the same as for dilated convolution. So for example, if you allow the transformer to always see like a fixed width, left and right, even that seems to help a bit. So seems like the transformers are like a good way to process these sequences, at least, in the abstract space. It could be because they maybe maybe they overfit less than convolutions. I don't know. I don't know the exact reason. And we also had some smaller improvements on pooling. So Basenji 2 uses pooling, max pooling. And we saw that by using attention pooling, you are probably losing less information because it's a bit more expensive operation where you say, how much, when you do your averaging of your nucleotides, you kind of weight them by attention. So it's quite nice because it allows you to kind of interpolate between average pooling and max pooling while being more general. So that was also one of the things we saw that helped.
I don't think I had seen the idea behind attention pooling before this. Is this an idea that you and your team came up with?
I mean, yes, but I later saw its other incarnations of this in the literature, so I don't want to claim the idea. But it made sense to me to have something more flexible than average pooling or max pooling, and also because attention is really good at aggregating information. I thought maybe it would be also good for pooling.
And that all makes sense to me, that a lot of these components in neural networks are extremely flexible, that you can change out all of the parts. People like using the parts that have been made before, like max pooling, average pooling, etc., when in fact everything is quite flexible.
Yeah, it's kind of stacking Lego blocks, and then maybe sometimes you come up with a new Lego block, and especially if you manage to improve the model, that's quite fun.
I have to say though that I didn't quite appreciate the extent to which things can be changed. The kind of two things, this is totally off topic by the way, but the two things that really helped me realize how flexible all this stuff can be. The first is this line of work from Kwok Lei about learning the operations. So basically they have this set of basic operations like additions, multiplications, etc. And they run evolutionary programming to just try to learn more complicated operations and activation functions from the data by like kind of composing these together and using genetic algorithms. And they've been able to rediscover not just some of the more commonly used approaches like transformers, batch normalization, etc., but also improve upon them. And so that really helped me think about how all of these operations can be broken down. All these layers can be broken down to their basic operations. And the second is actually switching to PyTorch. I was really skeptical that anything could be better than, Keras for the Theano backend. That's, kind of the state of the art in deep learning programming for me. But then once you switch over to PyTorch, and I guess this idea of having a computational graph that can be extremely flexible, you can change so much stuff. It kind of opens your world past the idea of using just the preset layers in something like Keras or the built-in TensorFlow stuff.
Yeah. I absolutely, I agree to both things. Yeah, I find the neural architecture search quite mind blowing. Although it's difficult to run for large models, so I guess you need big amounts of compute on this. In terms of frameworks, I think frameworks have gotten really good overall. There's one thing that people are really excited about in DeepMind is JAX, which is kind of an alternative to PyTorch. It's slightly, it's a bit different, but. It's basically like NumPy that runs on TPUs, and you're able to AutoGrad plus NumPy plus TPUs basically.
Yeah, I was basically, I have a outsider's history of TensorFlow, whereas you directly work for one of the companies there, so you probably have more accurate view of what's going on, whereas I have more hot takes. And it just seems like TensorFlow is kind of a hot mess. that there are so many different competing versions within TensorFlow of how to actually do things. And it seems like JAX is just the latest incarnation where like, well, everything we've done so far has been a nightmare. So let's make something that looks like PyTorch. Do you have any thoughts on the difference between JAX and PyTorch?
Yeah, I think the hardware is a key difference. Like TPUs require special compilers to run on them, and because they have a more restrictive architectures than GPUs, you really need the compiler heavy lifting in order to. So basically, you need the operation to be laid out ahead of time so that you can optimize them in order to execute them efficiently. Because like as soon as you start using more lower level, compute things, then you have to rely on your compilers to do all the heavy lifting. And because of that, you really have to, when running on TPUs, you really have to compile your program and you have to know ahead of time how it's going to execute. So that's why, like, if you were to run like just if you were to run eagerly on TPUs, it would be very slow, I think, and it will burn a lot of memory and so forth. And because of that, I think, you really have to, JIT your functions. Like you have to do just-in-time compilations. I don't know the exact history of I'm not the best person to ask about tensor programming. Why not just adapt PyTorch? But I think it has to do a bit with the compiler things and JITting. But yeah, I think I would redirect that question to other people at DeepMind and Google, because if any of my answers would provide injustice. Having said that, I find both JAX and I think all three frameworks have their own advantages. Although like if you're running a GPUs, probably PyTorch is really good. Because if you don't have to compile things, it's super useful to be able to run it eagerly. Whereas TensorFlow has a big ecosystem, which is part of it probably why it makes it difficult for people to why the complexity rises if you start to support too many things, which is something that JAX tries to avoid. It basically tries to be very minimal. To have to support as few features as possible, but as much as necessary.
Good to hear that you're fully endorsing PyTorch and telling people not to use anything else.
No, I'm not doing that. Basically, use whatever gets the job done, and so whatever you like and gets the job done. It's really cool that we have these frameworks nowadays, and that because like imagine where you had to like code up backpropagation yourself or write code I don't know in Lua not Lua in what was it in Lisp or whatever.
It was Lua,
the
original Torch. I remember when I started that the cafe was the thing that was out there, and the only way, like it only took command line operations and you had to specify your model in a text file.
I remember. I haven't used cafe, but I remember people complaining about the difficulty to tweak it if you wanted to tweak it.
Okay, well, at this point I'm kind of out of questions. Do you have anything else that you'd like to share about the project?
There's just one more analysis I wanted to highlight, and which is something I liked in the paper, was our analysis on eQTLs, which are variants associated with expression, and there one of the benchmarks was the task of distinguishing eQTLs that were statistically determined to be causal, because most eQTLs are not causal because of the co-inheritance between variants. And then the question is, can the model distinguish between variants that are deemed causal versus those that aren't? And we saw pretty good performance on that task, and also an improvement on this task. And I think this is a promising direction moving forward.
So just to be clear, these are variants where you have a whole bunch of candidate eQTLs.
Exactly.
And when you say the task of fine mapping, that it's trying to distinguish from all these candidate eQTLs, which ones are the actual causal drivers.
Exactly, yes. And there are Bayesian statistical methods. Like the field of fine mapping has improved a lot in the last years, and you have Bayesian tools that basically give you a posterior inclusion probability for every variant that tells you like what's the likelihood that this variant should be included in the final causal model. And we basically compiled a data set between variants that have high inclusion probability versus those that although they were originally in the EQTL set, they had a very low inclusion probability, meaning that they're very likely to be spurious correlations. And then the question is, can the model distinguish between these two classes of variants? And it can do, and it's helpful. This was also one of the recent David's papers where they have distinguished. They basically used the model to do the fine mapping task.
Yeah, I think that this idea of trying to do fine mapping is really interesting. Can you talk a little bit more, though, about where you get the set of candidate EQ2Ls from and where you get the set of actual causal drivers from? Because it sounds like you, at least for some of these settings, the true causal drivers, otherwise you couldn't evaluate the model.
Well, it's all still based on, statistical models of interpreting EQTLs, because like from a population study, you will only, you get associations, right? So basically the basic set of EQTLs is, I think it's used from GTEx data set where, you have, 1,000 individuals and you correlate their genotypes with expression across different tissues. And then you take that and you run statistical tools like SUSY. I forgot what the exact abbreviation stands for. And these tools take into consideration the co-inheritance pattern, so the linkage disequilibrium between variants, and the gene expression. And then they give you this Bayesian posterior inclusion probability that tells you like which one is likely to be causal. Although basically there are many loci where the method is not able to resolve it because there's just too much co-inheritance. And there are some regions where you are able to say based on this information. So, we only took the set that was kind of confident that, the method was fairly confident that, you're getting the right causal EQTLs. Although it's still a very challenging task to, exactly, So I guess it's up to down to probabilities that, on average, you're going to be right. Like many often, but not all will be causal that you called. Like we included posterior inclusion probabilities, which with 90%, meaning that if the method worked exactly as expected, then 10% would be, non-causal, whereas 90 would be causal.
That makes sense.
Yeah, but the set of variants was filtered quite heavily down from all the EQTLs because, as I said, from some loci, you cannot distinguish them and you cannot find them out, so you cannot evaluate the model there.
Yeah, I think that this task is particularly challenging because what you're saying is basically that you want to, that across a bunch of individuals, you identify variants that are correlated with the thing you care about, in this case, gene expression. But there are so many other things that can cause, association isn't causation. And there are so many things that can cause not spurious correlations, but can cause like more complicated independent structures like LD disequilibrium, etc. And so I do think that the idea of using machine learning models that are trained to predict something else like epigenomic state to try to identify like, does this variant change epigenomic state? And if it doesn't, then it's probably not changing gene expression. I think that idea makes a lot of sense.
Yeah, exactly. And this is kind of the fundamental way the model is classifying these two categories, because if the model says it's not changing any chromatin or anything, it's probably a spurious correlation.
Great. Well, thank you again for joining us. It's been great hearing more about the inner workings of this paper.
Yeah, thanks for inviting me, Jacob. By the way, this is a public endorsement. I really like your work as well. I think you've done some really good stuff. And look forward to see what's next for you coming out of Anshul's lab.
Well, thank you.
Yeah, it was a very fun chat. Thanks.